Okay, so for this lab, we're going to talk about how we grow bacteria and how we make a pure culture and why we need a pure culture. So when you look at cultures, this is a method of us multiplying these organisms by letting them reproduce in a culture media under very controlled laboratory methods using whatever temperature, pH, nutrients they need so they will grow. So these microbes rarely live by themselves, so the cultures are generally mixed with many different organisms. And this is especially true in medical settings and stuff like that. If you get a throat culture, you get something that has normal flora, you've got lots of different kinds of organisms growing. So the growing organisms require the proper nutrients, the proper temperature, the proper environment. We'll talk about that. And we need to prevent contamination. So we basically have to provide them everything they need to grow and in the proper environment. So typically this involves inoculating a sterile medium with a specific microbe and incubating it under controlled conditions. So some organisms like it warmer, some of them like less oxygen. So we will provide, based on where these specimens are coming from, the proper environment for the usual pathogens that occur. So in order to identify these organisms, they need to grow out in either solid or liquid media, and then they are tested using biochemical reactions to identify the organism. And then we can do an antibiotic sensitivity if something grows out. All right, so first of all, we need to provide nutrients. We have nutrient agar, which is solid, or nutrient broth, which is liquid. These are the fundamental ways that we grow these organisms, and they have this nutritional base that we make. Both of these are well-rounded diets for the bacteria they like. They provide them with a source of nitrogen, amino acids, peptides. There's usually some kind of yeast or beef extract that provides them with water-soluble vitamins, carbohydrates, salts, blah, 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 and then some amount of salt to maintain the osmotic equilibrium. Okay, so the liquid media is a broth. You can see it up there. I get a better picture. But it's a liquid media, and we call it a broth, and it is nutrient rich. It lacks these solidifying agents, agar, which comes from seaweed, is what is added. And it allows these microbes to multiply rapidly and uniformly, usually through the fluid. The most common one that we use is TSB, tryptic soy broth. It is a nutrient broth. It is a general purpose broth, and it's used to cultivate this wide variety. Anytime you have a general purpose media, whether it's an agar or a broth, it's going to grow most everything. Not everything, but most everything. We do have LB broth, lysogeny broth, which is standard for growing E. coli if we're doing molecular biology or plasmid DNA purification. And then we do have some selective broths where we put inhibitors in or indicators in. But basically, a selective one is going to select for a particular group of organisms by inhibiting the other guys. Okay, so we may put an antibiotic in or a salt or something like that, and that will suppress the growth of these other organisms. So this is a very typical TSB broth. This is how it comes in, and that's how it's used. Most of the time, we will be using solid media. When these specimens come in, a lot of times we'll have a tube of that liquid broth, and once we culture out everything, we'll stick the swab in a broth and see if anything else grows. Okay, so our solid media is gelatinous. It isn't jelly-like. I mean, you literally can pull it out of the agar plate and kind of flap it a little bit. It tears easily. It's very soft. and it's made from a polysaccharide mixture from the cell walls of red algae. Okay, and the main thing is it has agarose, which is a naturally gelling type polymer. And then it does have agropectin, but it ends up, you have a very jelly-like consistency that you work with, and I'll show you a picture. Now we have different types of augers. You can call them all kinds of different things. People have different names for them, but when you get a culture in, you're just not going to plate it out on one plate. And I'll explain why. But usually, we get a culture in from somewhere like a wound or something like that into the micro lab, and we generally plate it out on 5 or 6 plates because it helps us identify these organisms faster. Okay, if you just put it out on one plate, then everything grows, and if it's not all the same stuff, then you got to reculture it and reculture it to separate these organisms out. So generally what we do is we'll plate it out on several different augers. Okay, as I mentioned, we have this selective auger. This will inhibit or help the growth of certain organisms by using specific chemicals, and we'll look at some of these. We have differential augers, and that means you can tell the difference. So it has some kind of dye or characteristic change, color changes occurring, or growth patterns that change it so that we can actually look at the plate and differentiate just in these organisms. Maybe they're utilizing a certain sugar or something like that. I'll show you those. Supportive augers are those that support the growth of most organisms. So this is your TSA instead of TSB, tryptic soy broth. You're going to have tryptic soy agar. There are other supportive augers, but they grow most of these organisms. And then some guys are picky, what we call they're fastidious, and they have to be supplemented with high nutritious materials to allow for the growth of these picky organisms. And I'll give you an example in a few minutes. So here are the general types, okay? So we have a general purpose media nutrient auger. This would be the tryptic soy, as I mentioned. And then we have chocolate, which is a tryptic soy auger with sheep's blood that's been cooked a little bit. We have blood auger, which is nutrient auger with blood in it. We'll look at that. And then again, you cook it, it becomes chocolate. And then we have McConkey. This is selective for gram-negatives. We can have the Savarot dextrose, which loves fungi and yeast, and mannitol salt, which is selective for staph, and then EMB place, which like to grow gram-negatives. So, if you're plating out a specimen that's come down, you have no idea what's on that swab. So what we generally do is pick a nutrient auger. If it's coming from certain areas, maybe we're looking for Neisseria or something like that, they suspect it may be Neisseria, we would add a chocolate auger. But we usually have a TSA auger and sometimes a blood auger plate. And then if you look at it, we can pick one of the gram-negatives. And then there's another auger, CNA, that chooses for gram-positives. So, we can look at the plate, everything goes out on the nutrient auger, and then we can look at the McConkey auger. If that's blank, that means whatever's growing is gram-positive, and vice versa. So it helps us differentiate these quicker and get a pure culture, which we need to do antibiotic sensitivities, by the way, and identifications. So let's look at our tryptic soy. I'm going to go through these. Tryptic soy auger, very supportive, general media will grow most organisms. It's the same as the tryptic soy broth with auger added. It is the most commonly used auger. So you can see it. Somebody put their hand down on this auger. It is a very clear auger. basically transparent, very easy to pick up. It will grow fungi, it will grow yeast, it will grow gram negatives, it will grow gram positives. About the only thing it won't grow is some very, very, very fastidious organisms. So this is our most commonly used agar that we use in the micro lab. Now, blood agar, this is an enriched agar. It has 5 to 10% mammalian blood, not human. And it's used to grow some fastidious organisms and allows us, we'll talk about this when we get to strep and staph, it allows us to observe hemolytic patterns. So we can actually look at these blood cells and see if the blood cells are lysed. And that becomes important when you talk about streptococcus, and we'll get a whole lecture on this, and we'll look at this hemolysis patterns towards the end of the course. But, a beta strep, well, beta strep means that that red cell is completely lysed and the hemoglobin is completely degraded. And that tells us a lot about the organism. So using a blood agar will allow us to grow most of these guys, but at the same time, observe if there's a hemolytic pattern. Chocolate agar, as I mentioned, contains these lysed red blood cells. Cook them a little bit, and they release the nutrients for the fastidious organisms, primarily Haemophilus influenza, which causes influenza, and Neisseria gonorrhea, which causes gonorrhea. It does not contain chocolate. People say, so many students tell me, it's got chocolate in it. No, it doesn't have chocolate. It's just when blood has been heated, it turns brown and it looks like chocolate. All right. So those are enrichment agars. The McConkey, I showed you that is selective and differential. So you can have a selective and differential agar. We have several of those. It selects for gram negative. So it inhibits the gram positive bacteria. And then you can differentiate between these gram negative bacteria as to whether they can ferment lactose. So they will turn different colors. So you can say this is a gram negative lactose fermenter or a gram negative non-lactose fermenter. And that puts you in a category in terms of trying to identify these bacteria. Mannitol salt is selective, highly selective for Staphylococcus. Very high salt content. It inhibits most other bacteria. And this helps us identify our lovely little Staph aureus, which is really pathogenic. Okay. EMB eosin methylene blue. Okay, this is selective and differential. Eosin is a red dye. Methylene blue is a blue dye. So this selects for gram-negative organisms, and it has eosin Y, which is red, and methylene blue, and they will react with any acidic products resulting from lactose fermentation, and it colors the colonies. So lactose fermenters cause the precipitation of the dye on the surface of the colonies. And so if you get no fermentation, it's colorless, like right here. If you get a small amount of acid, it turns this kind of pink. If you have large amounts of acid, you get this beautiful green sheen on the plate. So it's pretty nice and interesting to look at. Savarot, as I mentioned, is a specialized media with a lower pH than antibiotics. It's designed to isolate fungi and yeast and molds. I mean, when we get a fungal culture in, this is what we're going to put it on because it'll do a real good job of growing these guys out. So again, when we work with these specimens, we will culture a submitted specimen on the appropriate media based on the diagnosis or the type of specimen presented to us, the area from which the specimen was obtained. It does make a difference. If this came from the OR and it's in the gut, and it may be an anaerobic bacteria, so we have to treat it as an anaerobe. So it all depends. I mean, I'm not going to look for certain bacteria in certain areas. I know what the normal flora is in some of these things, and so that's what we're going to do. So this does require usually plating on several plates, usually a supportive plate, and then selective and differential plates so that we can differentiate them out. And these plates are incubated at different temperatures and oxygen contents. So, a wound culture, we would usually, 5, 6, 7 plates. We'd put several at 37 degrees. Some bacteria like it a little cooler, so we would have a 30-degree incubator. Then we would, in case there was some kind of organism that didn't like too much oxygen, we'd put it in a what we call a CO2 incubator, which has more CO2 in it. So we can, anaerobes, we can put it in an anaerobic, remove basically the oxygen and stuff. So there are many different ways that we can do this, and this helps us, one, support the growth of that organism, and two, more quickly isolate the organism so that we can get a pure culture and then we can get the results to the doctor faster. Okay. So just FYI. Okay. So when we obtain pure cultures, we need to have a pure culture, nothing mixed in it, a pure culture of a single organism to, one, identify it, and two, to, if need be, do an antibiotic sensitivity on it. We have to have a pure culture. If we try and do an identification or sensitivity on it, it's screwed up. It is worthless, okay? So the way we do that is we try and get a pure culture. This is a culture composed of cells arising from a single progenitor. So we call this a colony forming unit. So we're looking for a single colony on the plate, something that we can go in and pick out a single colony, because everybody in that colony comes from the same beginning. All right. So we use aseptic technique to prevent the contamination of these things, and we have 3 different ways to do it: streak plates, pour plates, or and spray plates. Usually in the microbiology lab, we're going to be using a streak plate because it's very quick and very fast. So we take the original culture, we directly go in with a loop, sterilize the loop. We pick up a small amount of that colony, and basically the whole idea now is to streak it out, thin it out, and make sure, whatever we've got from the original colony, what we want to do is pick up one individual colony. So if I got a swab, I'm going to swab that plate all over, but I'm going to do it in a special means called 4-quadrant streak so that we hopefully pick up just one colony. This is the most common. So the way we do this is we take our original specimen, okay? Sometimes if that may be the heated, or we may just streak. So like if we got a swab or we got a liquid, we put a drop there, we got a swab, that would be the first streak. We go across 1 quadrant, okay? And then what we do is we'd sterilize our loop and then streak it across this. And you only go across this part one time. Then when you come back, you don't come back into here. And then you do it another time. And finally this, okay? If you're trying to just isolate something that you're picking up, then you're going to do the inoculating loop and go and thing. But it's basically you are doing, if you were in a lab, you would be doing this. And what ends up is you get something like this. So this is obviously, okay, this is your first streak. It is loaded down in bacteria. This is quadrant 2, quadrant 3, and your little wiggly line. And you can see as you start to spread this stuff out across the plate, you start to get individual little colonies by the time you get to the fourth quadrant. And you can work with this. This is one colony that you can work with to determine your organism and your sensitivity. Okay. You can use pour plates. Not very often are we going to do it, but we do use it in research and stuff like that. It is a common procedure to make cereal dilutions and then put them out on a plate. It's not something we use very often, but we will take that and we'll spread it evenly and you get the results. And this is the problem, is it may not be, you'll put only a little bit. It doesn't necessarily guarantee that you're going to get isolation. So you can do that, but you can actually allows you to count colonies and growths when you're doing research and that sort of thing. You can do a cereal dilution. Just keep diluting until you get down to a couple colonies, okay? So that is it. That is how we basically make a pure culture. And again, we need the pure culture. We'll talk about this when we need to identify the organism and do an antibiotic sensitivity. Okay, so that's it for pure cultures.